We're delighted you're here, and for those of you who are joining us through streaming, we're glad to have you here as well. And I know our participants like having an audience. We did this last year, basically virtually, and you lose a little bit, but it was still good. I think this is probably my favorite event. It's such a great treat to see how talented our students are and what a difficult challenge they face. Realize what you have to do is somehow distill all the work that you've done, that you've to a professional degree, and be able to tell a lay audience about it in 3 minutes with a single static slide. That's a real skill, and so we really appreciate the work and the interest they do in performing here. Actually, the 3MT began in 2008 at the University of Queensland in Australia. The next year, a number of universities in Australia and New Zealand picked it up. Today, there are over 350 universities in over 18 countries who have 3-minute thesis competitions, 3MT. Actually, the recipient, the winner of today's competition, will go on and be a contestant for us at the regional meetings. And someday I'm pretty sure we'll probably have a national, but right now it's regional and local. So it's really a great event. And it also gives the university an opportunity to showcase the really exciting research that's going on here and the fact that graduate students participate, not only participate in it, but actually lead much of the research. So I'm going to turn this over now to our Executive Director, Dr. Jill of Center for Graduate Life and Learning, Dr. Jill Huerta, who will continue on with this program. Jill.
Well, good afternoon to those of you who are joining us in person here, and to those of you joining the live stream, and good afternoon, finalists. Really? Good afternoon, finalists. You're about to present. You have to get your blood moving a little. As Dean Reynolds just described, 3-minute thesis is about training graduate students to explain highly complex technical information to professionals who are not necessarily in their field, but to professionals who need that information to make impactful decisions. It's a critical skill. And because of this, every year at the 3MT finals, we try to recruit a panel of judges who do just that in their daily lives. They take in complex technical information and use it to make impactful decisions. And today's panel of judges fits that bill precisely. We have people from the private sector, people from nonprofit sector, and the government sector here. And I'd like to take a minute to introduce them. So I will go in the order that you all are sitting in this row right here, and perhaps you could wave as I introduce you. First of all, from my left, we have Toby Holmes, who is Planning and Development Director at University City Partners. University City Partners is a nonprofit that promotes investment and development in the University City area where our campus is located. So can I get a wave, Toby? Next, we have Janelle Cotto, who is Senior Human Resources Manager at Geldwen. And Geldwen is a door and window producer headquartered in Charlotte, but doing operations in what, about 19 countries, Janelle? 23 countries. So a large company that has been a fantastic partner with the Center for Graduate Life and Learning in our Accelerate to Industry program. So several times company representatives have come in and provided professional development for our graduate students, which we appreciate. So if you could give a wave. Sitting next to Janelle, we have Victoria Watlington, who is a City of Charlotte Council member, District 3. and also a service operations manager for Coca-Cola, and a Charlotte alum from engineering. So great to have you here. Can you give a wave, Victoria? And finally, next to Victoria, we have Senator Mujtaba Mohammed, who is back again after judging 3MT in 2019, the last time we were able to do this face-to-face. Senator Mohammed is a North Carolina state senator. He's also assistant public defender for Mecklenburg County and also a UNC Charlotte alum, having received his bachelor's degree in an area near and dear to my heart, history, and then going on for a law degree at North Carolina Central University. So thank you so much, judges, for taking the time to be here with us today. There are a few other people that I'm pleased to have the opportunity to thank, and I have quite a list here. First of all, several people volunteered their time to coach our finalists today. Catherine Hall-Hertel, Elise Demeter, Patrick Madsen, Laura Smales, and Stephanie Norinder. Also, there were 20 people or so, internal staff at Charlotte, who served as judges in the preliminary round. Too many to name, but we appreciate their effort as well. Natasha Strassener and her team have made this streaming possible, even though we didn't make life easy for them because we wanted to be in this room. So they brought their equipment on the road and agreed to stream this for us today. So thank you, Natasha and team. And a big thank you to Miguel Wilson, who is our graduate assistant at the Center for Graduate Life and Learning, who worked hard to make the organization and logistics of this event go well, and also brought so much enthusiasm to the recruiting process that he helped us to recruit a record-breaking number of grad students participating in the preliminary rounds. We had a third more than any other year this year. So, and partly thanks to his efforts. So now I'm pleased to introduce our MC for this event, who is Dean Adams. Dean Adams, yes. Dean Adams, and Dean is his first name, it's not a title. But he is also Associate Dean of Performing Arts Services for our own College of Art and Architecture. And before that, he was founding artistic director of the Connecticut Theater Festival in Simsbury, Connecticut. As a producer and director, Dean has presented or co-produced productions with some pretty impressive groups that include the Parsons Dance Company, Palalabus, the National Theater of the Deaf, and the Emerson String Quartet. And he also designed and directed a touring production of Once Upon a Mattress, which was the first musical event to tour the People's Republic of China in 1987. Dean, thanks so much for being with us today, and I will hand it over to you.
Thank you. It's wonderful to be here, and yes, it's Dean, never gets old. Welcome to 3MT. It's the academics' answer to the elevator pitch. And so we're looking forward to this. So here are the rules. The rules of 3MT state that participants may use one single static slide with no animation, no props, and no additional media. Presentations are limited to 3 minutes maximum, and any competitor exceeding 3 minutes will be disqualified. Contestants, when you have 30 seconds remaining, Alberta in the front row will hold up a yellow card to let you know. That's 30 seconds. Thank you. Okay, when your time is up, she will hold up a red card. It's a little bit like soccer, isn't it? You don't want a red card. Okay, so our judges will score and participate, and the judge's decision will be final. So our participants will be judged on 2 criteria. Number 1, comprehension and content. Did the presentation follow a clear and logical sequence? Was the topic, key results, and research significance and outcomes communicated in a language appropriate to a non-specialist audience? Number 2, engagement and communication. Did the presenter capture and maintain their audience's attention? Did the slide enhance the presentation? So, prizes will be awarded on the following. First place will receive $750 and travel funding to compete at the regional 3MT competition, which you just heard about, in Raleigh, North Carolina in February of 2022. Second place will receive $400 and third place will receive $300. We will also ask the audience to vote on a People's Choice Award once all the participants have presented. So as you're watching, whether here or at home, keep in mind which presentations stand out so that you can vote for your favorite at the end. There will be no cash prize for this award, but the winner will receive a certificate and bragging rights as being the People's Choice. We will now begin the presentations. So our first contestant is Shanique Ashrafi, and from the Infrastructure and Environmental Systems Engineering PhD program.
Good afternoon, everyone. Did you know that in our current society, we spend more than 90% of our time indoors? This makes the indoor environmental qualities have a significant influence on our health condition. The building sector consumes 40% of globally produced energy to provide us with comfort and health, but we are still largely dissatisfied with our thermal comfort condition. How many of you have the experience of working in a freezing office environment? Have you ever wondered why building managers are willing to pay more for the electricity bills to make buildings so cold in the summertime? Maybe my answer will surprise you. This is simply because the thermostat temperature in office buildings are set based on standard codes, which are pretty old and are developed mostly based on male occupants' thermal comfort. So the problem here is generalized standards, which makes it impossible to consider our differences in our thermal sensation based on our own age, gender, race, metabolic rate, or clothing level. This has caused a common problem, which is called sick building syndrome. Basically, our buildings are making us sick. But imagine a controlling system that is working on providing comfort for the current occupants of the building based on their own preferences. Pretty amazing, right? This is what my research is about. We are working on using the feedback from the occupants to control the thermostat temperature. But how are we going to get the feedback from the occupants? Our system is a totally non-intrusive platform that is capable of predicting your thermal comfort based on your facial skin temperature. So as you can see in this control loop, first, the system detects the faces of the occupants in the room. Then, through our dual camera system consisting of a thermal and RGB camera, we can have precise temperature readings from different facial areas in our face. Then the temperature data goes into the cloud as an input for our AI-based prediction algorithm to predict the group thermal comfort of people in this room. But don't think that we have forgot the energy considerations. The outcome of this platform is a thermostat temperature that is capable of creating a balance between thermal comfort and energy saving at every point of time. So, don't you think that considering diversity in controlling our environmental systems would be a great addition to the future of our smart buildings? Thank you so much, everyone.
All set? Okay, good. Our next contestant is David Brown from the Bioinformatics PhD program.
You might have heard the phrase antibiotic resistance crisis. Disease-causing bacteria are surviving our antibiotic treatments more and more often, leading to longer, deadlier sicknesses and higher healthcare costs. According to the World Health Organization, 700,000 people die each year because of antibiotic-resistant diseases. That number is only projected to increase. This is the antibiotic resistance crisis. My research is focused on helping us escape that crisis by understanding the past to predict the shape of things to come. Many antibiotics work by fitting small pieces of bacteria called targets. For our purposes, that toy ball on the left represents a single bacterial cell. Each hole is a target, and each yellow shape is an antibiotic. A square antibiotic fits a square target, and the bacterium is killed. A few bacteria will always survive antibiotic treatments because of small differences in their DNA. These changes are passed down to the next generation. Again, in our example, any bacterial cell without a square target will survive and pass on those genes. Over time, these generational changes lead to groups of bacteria which resist antibiotics. These groups can also be thought of as families. Bacterial families survive through these processes of change and survival, evading our antibiotics. So how do we escape the antibiotic-resistant crisis caused by these families? A great way to understand families is by looking at the past through a family tree. You might have seen a family tree before when you submit your DNA to a group like 23andMe. An example family tree is seen at right, where a square changes into different shapes through small changes in each generation. A family tree is a tree built on genes. We call that a phylogenetic tree. Phylogenetic trees can tell us how bacteria's antibiotic resistance genes change over time. My work uses both phylogenetics and computing to look at bacterial genes and tell us what is coming next. I can examine what they have in the past and what they have right now. I can look at their antibiotic resistance genes to see what weaknesses we can exploit. Understanding the phylogenetic history of a bacteria can help us make predictions about what comes next. Predicting what comes next can help us escape this crisis and save lives. Doctors and hospitals can use my research to make better choices about antibiotic use. That's how we shape up against bacteria. They change through generations, just like our own families do. You might have your grandfather's nose or your mother's eyes, little marks of the past and the present. Bacteria have hints like those too, and they can tell us what is coming next. We have to be smart to preserve our antibiotics. With my work, we'll be in good shape.
Judges, are we ready? All right. Our next contestant is Varsha Goodikindi from the Nanoscale Science PhD program.
What if I told you that we are slowly moving to a future where a single paper cut can become untreatable if it gets infected with a resistant bacterial species? I know what you guys are thinking. Another pandemic? Is it bacteria this time? Where are the antibiotics? It's true that antibiotics were considered the option when treating bacterial infections. They kill bacteria by following a specific pathway. However, we humans have overused antibiotics to such an extent that bacteria has now evolved to block all these paths. Multiple bacterial strains exist now which can block all the paths an antibiotic can take to kill it. They basically become immune to most drugs, and they have been termed as multi-drug resistant bacteria. Clearly, antibiotics are not the answers, and researchers are looking for other options. Silver is a very interesting candidate to do this. Ancient Egyptians used to consume and store food in silver containers to avoid bacterial growth. We now know that they did this because silver is able to gradually release silver ions, which can kill bacteria by multiple pathways. I'm using silver, but a form called as nanosilver. Such a small size allows better interaction with the micro-sized bacteria. In my research, I am designing nanosilver with specific coatings that can release silver ions, but in a controlled manner. How do I do this? Visualize a sponge. You put some soap on it, you put some water on it, and you squeeze it. You see a lot of foam. The more you squeeze, the more foam you see. The nanosilver which I have designed work in a similar manner, but I'm using more light to squeeze out more silver ions. The light exposure activates the coating present on the surface of nanosilver to release a very radical form of oxygen, which can accelerate the release of silver ions. More light, more silver ions. The idea is not just to increase the release of silver ions, but to do it in a controlled manner. This way, lower dosage of silver can be effectively used to release more silver ions and can amplify their ability to kill the resisting bacterial species. Right now, every year, 700,000 people are fatally affected by these resistant bacterial species. Experts say this number is going to rise. This research is focused on reducing that number and making death by bacteria ancient history. Thank you.
Our next contestant is Shanique Lee from the Curriculum and Instruction PhD program.
Have you ever had a teacher who understood you like no one else? They had a way of making you feel seen and validated and really empowered to learn. Or what if I told you that a lot of Black women teachers have a natural ability to do this, especially for Black students who are most often at the center of education reform initiatives? Well, it's true. A lot of them, whom I call critical Black women teachers, are part of a long legacy of classroom activism and achievement. But the problem is, today, Black women make up only 5% of the teaching force and have had consistently high rates of attrition ever since schools were desegregated in 1954. So the purpose of my dissertation was to contextualize this attrition in order to better understand how to support Black women teachers in urban schools. I wanted to honor the culture of Black women and create an authentic space for them to openly share their experiences. So I used Sister Circle methodology, which is a qualitative approach for Black women to study Black women. Its primary method of data collection is Sister Circles, which are affinity spaces to engage in critical group dialogue. And through these individual interviews and written reflections, 3 themes revealed the complexity of their struggle between loving and leading the classroom. To summarize my findings, all 15 participants really valued the magic that they created in their classroom, but many of them were worn down by the constant pushback they received against their teaching practices. This, along with other institutional and interpersonal issues, caused problems with a lot of their mental, physical, and emotional health, which made them feel unfairly forced to choose between pursuing their liberation or that of their students. Throughout all of our conversations, a lot of tears were shed, and many of the participants described leaving the classroom as the hardest decision they ever had to make. So we all need to get involved because these women are quite possibly the key to finally achieving educational equity. And here are a few suggestions. First, education policymakers should apply a critical race perspective in order to identify and eliminate those structural barriers to the work of critical Black women teachers. Second, teacher preparation programs can help equip their Black women teacher candidates to be able to anticipate and resist pushback by helping them to develop radical self-care practices. And finally, urban school administrators should create school cultures that welcome and support liberatory teaching practices. Ultimately, my findings reiterate why I chose to focus on sustainability in the first place. Because while retention aims to keep teachers in the profession, sustaining Black women teachers calls on the entire education system to become more welcoming and supportive. And instead of pushing them out, inviting critical Black women teachers in and helping to cultivate their professional practices over time. Thank you.
Our next contestant is JeanMarie Linker from the Masters in Bioinformatics program.
The noise that comes with our unprecedented access to information these days can make it very difficult to sift meaning from all the data. We need sound statistical models to help us do that. Now, we're all familiar with times that the statistical models have failed, sometimes spectacularly, in the financial markets, in presidential polls, in, turns out, predicting the success of Hollywood movies. There's a lot of money being made right now in Hollywood using statistical models to produce and promote movies. You know, in the early 2000s, the movie Charlie's Angels Full Throttle came out, and the statistical models predicted that it would be a huge success, but it was not. The statisticians that were responsible for that prediction decided that their models were missing a key feature, which was word of mouth, so they reworked their models to include that factor, and along came Snakes on a Plane. Now, Snakes on a Plane had everything. It had a stellar cast, a truly horrifying premise, and a lot of word of mouth. The models predicted that it would be a huge success, but it was not. So the point that I'm trying to make here is that sometimes statistical models that are very useful for existing data can fail completely in the face of new circumstances. And our attempts to fix those models or adjust them by including new factors can also open up avenues for further error. Obviously, I'm not doing my research in the field of Hollywood movies. I'm doing my research in the field of metatranscriptomics. This is the study of how genes express themselves in real-world environments. For example, if you look at the human gut microbiome, we might take samples from healthy individuals and samples from individuals who suffer from irritable bowel syndrome and use statistical models to answer questions like, what are the organisms that are present in those samples? And is there a difference in abundance or expression of genes between the healthy individuals and the unhealthy individuals? And here's where the problem is. The models that we're using to do that, the statistical models that we're currently using, were originally designed for another purpose. They were designed to look at differences in expression for an individual organism across multiple environments. And now we're looking at many organisms across multiple environments. So in my research, I am working with both data from the natural world, in fact, human gut data, and also simulated data that's simulated in a lab, to see if these models are holding up. Are there factors that we need to consider? Maybe we need to scrap our models and start over from scratch. This is important because metatranscriptomics and the sister fields of metagenomics and metabolomics are burgeoning sciences that are helping us to understand our environment and human health. We cannot afford to make a snakes on a plane style mistake in these fields. Thank you.
All right, our next contestant is Ritika Malik from the Bioinformatics PhD program.
Cancer. The grim reality of this disease is that everyone in this room knows at least one person who has suffered from or lost their lives to cancer. Cancer is a monster. It has been treated as a disease, but we are looking at it genetically and treating cancer as an evolutionary process. One of the things that cancer is really good at is evading the immune response of our body and growing silently inside us. Cancer grows inside us in different types of ways, through tumor generations or through mutations. But what I'm studying is, why aren't these cancers eliminated despite us having such a robust immune system? How does cancer evade detection by the guardians of our body, the macrophages? Turns out, cancer has evolved and is now co-opting our own bodies. The way it does this is by co-opting a signal in the body, which helps us detecting our own cells from foreign cells. So these signals, operated by the signal regulatory proteins or SERPs, are used by cancer cells to trick the macrophages by giving off a "don't eat me" sign so that cancer can grow on uncontrollably and undetected inside us. The modern day genomics has given us a way to understand the molecular machinery of cancer. All of the genes that cancer uses have evolved over thousands and millions of years, and studying those genes in animals like dogs gives us a fair idea of how human cancers work. Also sheds perspective into why some animals like elephants do not have cancer, and maybe we can engineer that part in order to combat human cancer. I have made this comprehensive database of SERPs from all the animals that have been sequenced till date, and this database will only grow as we get more and more data from our collaborators at the NC State Museum and University of New Mexico. From this modern data, I want to regenerate the history of SERPs, understand its origins better, and detect what is functionally closest to humans. Here you can see the SERPs in blue were the ones previously identified, and the ones in red are the ones that we have identified recently. From my research, I'm making this evolutionary roadmap that can be used to guide new drugs, design new clinical trials, have more drug designing, more therapy designing for people with different genetic backgrounds. We can come up with something completely novel and help in the aid of generation of personalized medicine. I believe tackling cancer genetically from the roots can finally help us win the war over cancer for good. Thank you.
All right, our next contestant is Keith Murphy from the Health Services Research PhD program.
Whenever we are presented with an image or a picture, it's usually a fairly simple task for us to identify what it depicts. For example, if I showed everyone here the image in the top left, I don't think it would come as a surprise to any of you for me to reveal that this is an image of a cat. But what about this image makes us think that it's a cat? The pointy ears, the fur, the whiskers. Your brain is coming up with all of these key features that are allowing you to conclude that this is an image of a cat. Now, my research is focused on training computers to classify images using these key features. However, a computer can't think about images in the exact same way as our brains. In the computer's case, it breaks the image of the cat down into tens of thousands of pixels, finds out what features are most important from among these pixels, and then concludes that this is an image of a cat. A picture truly is worth 1,000 words. Now, modern medicine relies heavily on these images and pictures, which we normally think about as CT scans, X-rays, MRIs, and so forth, to do such things as predict whether or not we're going to need a procedure, if we're going to go on to develop a cancer, or the whole host of other possibilities that could be tied to our health. I thought that this field represented a great opportunity to test out these artificially intelligent, or AI machines, and try to answer some of these questions in medicine. In my own case, I was concerned with predicting whether or not a patient would require a simple or a complex hernia procedure. I based the AI model against what is considered the gold standard in diagnosing the necessary procedure in the patient, the surgeon. The surgeon utilizes his or her years of clinical experience to determine the patient's treatment plan based on these image inputs. So what I've provided for you today is an example for how we can mimic the surgeon's decision process by using the AI model. In this case, the AI model takes our image inputs, which is the CT scan, and finds out what pixels and features are most important to our outcome of interest, the difficult hernia procedure. So why does this research matter? Well, if an individual gets a difficult hernia procedure and doesn't need it, then that's a huge physical burden that you've placed upon that patient. Conversely, if a patient requires the difficult hernia procedure but never gets it, then in 3 months, that patient is going to have to return to the hospital for a much more severe procedure with a much higher probability of complications. My PhD research is centered around combating and reducing these two life-altering scenarios. So be careful. The next time you go to the doctors, they might be a little bit more robotic than you might expect. Thank you.
All right, our next contestant is Amanda Sargent from the Organizational Science PhD program.
It's not what you know, it's who you know. Our parents have said it, our teachers have said it, and maybe we've even said it to explain how important networking is to getting a job or getting ahead. But what is it about who we know that gives us that special boost? Well, my research is examining a particular practice in organizations related to how who we know can help or hurt our workplace success: employee referral programs. Employee referral programs incentivize current employees to recommend someone from outside an organization for an open job role. And I think these practices may be accidentally introducing race and gender bias into hiring. Here's why. We know from prior research that applicant names at the top of a resume can lead hiring managers to infer the race and gender of that applicant. And that in turn, these inferences can trigger implicit biases that lead hiring managers to evaluate equally qualified candidates differently, often resulting in women and people of color being evaluated less favorably compared to men and white applicants. So if an applicant's name on the top of a resume can lead to these different evaluations, might not also the name of the referring employee, which is usually also visible on an application, do the same thing? I think it can. So to test this idea, I'm running an experiment where I'm asking experienced hiring managers to evaluate the resumes of 4 hypothetical job applicants. Now, I've removed the names of the applicants from the resumes, and while all the resumes look different, they're actually the same in terms of quality. The only difference between the resumes is the name of the referring employee, listed at the top of the page and selected to signal that this person is either a white man, a black man, a white woman, or black woman. Now, this research is still in progress, but I've conducted a pilot test, and here's what I found so far. While all of the applicants were evaluated essentially the same in terms of competence and commitment, certain applicants were more likely to be recommended for interviews than others, based on the race and gender of the person that referred them. So people referred by white individuals had the highest recommendations for interviews, while people referred by black women specifically had the lowest. Now, what these findings suggest is that what matters about who you know, at least in employee referral programs, may just be the race and gender of the person that referred you. And if this is the case, these programs are introducing bias into hiring, but there's hope. When we identify sites of inequality in organizations, we can begin to strategize solutions. My research can help organizations evaluate their practices and take action to ensure that all people are given an equal shot at getting the job and getting ahead. Thank you.
Okay. Our next contestant is Kelsey Sycon from the Masters in Civil Engineering program.
Imagine sitting down for dinner and receiving this well-balanced meal. You've got pasta, veggies, fruits, and for dessert, a credit card. Now, I'm sure none of us would chomp down on that credit card, but in reality, we are surrounded by plastics, from grocery bags to our clothing and carpets. These all physically break down, allowing tiny pieces of plastics to end up in the air we breathe, the water we drink, and the food we eat, to the point that we could each be ingesting a credit card of plastic every week. Now, plastics cycle easily from laundry and showers and toilet water in our homes to collect in wastewater treatment plants. During treatment, solids are separated from water, then sent out to farmers for use as fertilizer. The treated water is sent to nearby streams, which may later be our tap water. So we are recycling plastics, just not in the healthiest sense. It is crucial to break this cycle and prevent the release of plastics from treatment plants. To do so, we must first analyze the quantity and type of plastics in the wastewater, in treated water, and the solid byproducts. My lab does just that. We isolate the plastics from the wastewater samples and trap them on filters for analysis. You can see the brightly colored plastics on the filter in the middle photo. Second, it is critical to characterize the effectiveness of individual treatment stages. This can highlight which areas may need some improvement. Whether it's an ineffective stage or may have a product that needs further treatment or proper disposal. For example, we've seen that the initial stages of treatment, which is removing the solids from liquids, also has the greatest removal rate of plastics. So if we know the solids have the greatest concentration of plastics, then we know to focus our efforts on treating the solids and managing them. My analyses can guide plant operators. My analysis can guide plant operators to improve plastic removal and disposal options.
Our next contestant is Samantha Taylor from the Masters in the Bioinformatics program.
Think of the last time you brushed your teeth. Did you brush your tongue? If you did, you may have noticed that your tongue might have been covered in a thin white layer of gunk. Lots of people's tongues appear whitish. It can be caused by drinking alcohol, dehydration, or even just being a mouth breather. It could also be thrush, commonly known as an oral yeast infection. Thrush is common in babies and older adults, but also in those with weakened immune systems. Thrush is just one of the most common pathogens that can be found in the saliva and cheek cells of our mouths, along with hepatitis C, herpes, HIV, the flu, and even COVID-19. Currently, to detect viruses, yeasts, and bacteria alike, biologists use PCR, which is essentially a molecular copying machine. PCR copies a pathogen over and over until it is detectable, but this method takes time to set up, hours to run, and even longer to analyze. Because of this, my research is a proof of concept project that uses a more pathogen-specific method of detection. Lamp-mediated isothermal amplification, LAMP for short, adds a saliva or cheek cell sample to a mixture of pathogen-specific chemical reagents. This concoction is then put on a heating block for 30 minutes, at which point it either stays pink in color or turns yellow, given that the pathogen is present. LAMP has a colorimetric approach, meaning that it can detect something with the aid of a color chemical. This can be used especially in nursing homes, where
to be used in nursing homes, especially, to cut out the middleman so that they don't need to send out the samples or take the patients into the doctors. A test that only uses an oral sample, a pre-made chemical mixture, and a heating block could also be used in situations where resources are limited, such as countries that are still developing, and in situations where a rapid test has not yet been developed or has a poor detection track record. LAMP is just as efficient and has the same sensitivity as PCR and is a simple and rapid amplification method that could be used on any oral sample if given the chance.
Final contestant today is Farida Yasmin from the Biological Sciences PhD program.
Let me start with a question. Raise your hand if you eat any of the following: tofu, milk, yogurt, instant noodles, dumplings, or ice cream. I see a lot of hands here. Are you surprised to know that these products all have soy in them? It's not just soy milk and tofu. This list goes on and on. As you can tell from all the raised hands in this room and all the products I listed, soy touches our lives every day. Now, I don't want to make you all too hungry before dinner time. So, let's embark into the wild and talk about the genetics behind soy. Plants are natural chemists. I study wild soybean because it offers higher genetic diversity. The soybean we consume has lost many important genes as a result of farming and agricultural practices. Due to less genetic diversity in soybean crops, they are not well equipped to fight against a variety of pathogens and pests. So, why do we care about this? These pathogens are responsible for billions of dollars in US soybean production losses. In my research, I found an important gene that produces a vital compound named soy saponin. And this gene is only found in wild soybean. This compound protects soybean and other legume species such as beans and peanuts and beans from pathogens. Not only does this compound help plants, but it can also help to strengthen our immunity. Using this knowledge, I'm going to apply gene editing technologies and breeding to make soybean and other crops stronger. This will reduce pesticide use, help us grow biofortified crop, and produce plant-based medicines to ensure plant and human health for the future. Clearly, making wild soybean our savior. Thank you.
Before I get to the long-awaited moment, I want to congratulate all of the finalists. This is not an easy thing to do, right? If you look at different polls that people take, public speaking is one of the very scariest things that people do. It's one of the most common fears. And you all have put yourself in this situation of doing something that's very difficult, and you've chosen to take a skill while you're in grad school and try it and leave your comfort zone and practice it while you're here. in this safe environment where you have lots of support and you can perfect it. So when you go out on the job market and you're interviewing and you have to talk to someone in human resources and distill your research down into something understandable, you've done it before. You've practiced it. So I can tell you how proud we are of you and how well you've done. So congratulations to all of you. All right, now we have the envelope. And I would like to begin announcing the winners. The first winner I'll announce is the People's Choice Award, and that one goes to Kelsey Sycon. You want to come up? We have a certificate. Thank you. Congratulations. Next, we will go to third place, and third place goes to Shanique Lee. Congratulations, Shanique. Nice job. Next, we have second place, which goes to Farida Yasmin. Congratulations, Farida. Finally, we're ready to announce first place. As you recall, the first place winner gets a cash prize like the other winners, but this person also will get an all-expense trip paid to Raleigh, North Carolina to compete at the next level in the regional competition at the Conference of Southern Graduate Schools annual meeting in February. And this honor of representing UNC Charlotte at that next round goes to Amanda Sargent. Congratulations, wonderful job. So this closes our 3-minute thesis competition. We invite you all to join us in the reception in the lobby, have a glass of wine or an iced tea and some appetizers and kick your weekend off, especially the finalists. Thank you.